Hello friends, in previous video I explained about cell structure. Cells are the basic building blocks of all living things. The human body is composed of trillions of cells. They provide structure for the body, take in nutrients from food, convert those nutrients into energy and carry out specialized functions. Cells have many parts, each with a different function. Today I am going to explain about isolation of biomolecules and various products obtained by subsequent centrifugation. Objectives: on completion of this lesson, you would be able to know isolation of biomolecules, various products obtained by subsequent centrifugation. Experimental approach in biochemical studies. Studies on cell organelles and biomolecules have provided lot of information about their functions and metabolism. The three main steps or approaches involved in these studies area: A. Isolation of organelle or biomolecule. B. Determination of their structure. C. Analysis of their function and metabolism. A. Isolation of cell organelle or biomolecule. For this, the cell has to be broken down, distracted. Later, the organelle or the biomolecule has to be separated or fractionated. The two processes involved in their isolation are: 1. Homogenization and 2. Centrifugation. Homogenization: it is breaking of the cells in order to liberate the organelles or biomolecules. The cell's microbial or tissues, liver, are homogenized, usually with a motor driven pestle fitted in a glass tube. This breaks the cells. The organelles are extracted with a suitable buffer, TRIS, HCl buffer. The extraction of the organelles results in a suspension called homogenate. It contains all the intact organelles. Second, centrifugation. The next step is the fractionation of individual organelles like nucleus, mitochondrion, etc. For this, the homogenate obtained in the above step is centrifuged 3 times successively. It is called as differential centrifugation. It is done as follows. The homogenate is first centrifuged at a slow speed 600 G. It results in a pellet. Example, sediment settling at the bottom, and a supernatant, e.g. a liquid column above the planet. The pellet contains the nucleus. Second is the supernatant obtained as above is again centrifuged at a moderate speed 15,000 G. It results in a pellet and a supernatant. The pellet contains mitochondria. The supernatant obtained in the second step is again centrifuged, third centrifugation, at a very high speed 1,05,000 G. The pellet formed contains the microsomes. The supernatant contains the cytosol. The presence of a particular organelle can be identified by the appropriate marker discussed above. Further purification and isolation of the organelle can be done by using advanced technique like thin layer chromatography, gas liquid chromatography, and electrophoresis. B. Determination of structure. The structure of the organelle or biomolecule isolated is determined by various techniques. They are elemental analysis, spectroscopy, ultraviolet IR and NMR, mass spectrometry, X-ray crystallography. C. Function and metabolism. The function or metabolism of these organelles or biomolecules are studied by making use of intact animals, whole cells, tissue homogenates, isolated cell organelles, metabolites, and enzymes. Next is isolation of biomolecules. Extraction of DNA, RNA, and protein is the basic method used in molecular biology. These biomolecules can be isolated from any biological material for subsequent downstream processes, analytical or preparative purpose. In the past, the process of extraction and purification of nucleic acids used to be complicated, time-consuming, labor-intensive, and limited in terms of overall throughput. Currently, there are many specialized methods that can be used to extract pure biomolecules, such as solution-based and column-based protocols. Manual method has certainly come a long way over time with various commercial offerings which included complete kits containing most of the components needed to isolate nucleic acid, but most of them require repeated centrifugation steps followed by removal of supernatant depending on the types of specimen and additional mechanical treatments. Automated systems designed for medium to large laboratories have grown in demand. Over recent years, it is an alternative to labor-intensive manual methods. In the technology should allow a high throughput of samples, the yield purity, reproducibility, and scalability of the biomolecules, as well as the speed, accuracy, and reliability of the assay should be maximal while minimizing the risk of cross-contamination. Next is homogenization is a produce procedure where the fat globules in milk are separated into a littler, progressively predictable size to circulate them equitably all through the milk. At the point when milk isn't homogenized, the fat, which is less thick than milk, moves to the surface and sits over the milk as a layer of cream. Using synthetic concoctions during this procedure is bad for health. Before the homogenization procedure was utilized, milk was blended to accomplish an even distribution in its taste and texture. The homogenization procedure includes cutting down the size of the fat globules into minuscule parts that are scattered equably all through the milk. Homogenization normally is accomplished by siphoning milk through little opening under exceptionally high pressure. Milk is an emulsion with fat globules are scattered in a sisal skim milk manner. On the off chance that raw milk was left to settle, notwithstanding the fat would swell and retain the shape of a cream layer, and retain the shape of a cream layer. Homogenization is the treatment of the fat globules in milk achieved by passing it under exceptionally high pressure through a little hollow, which results in a reduction in the usual breadth of the fat globules. The net outcome, if we decided to be realistic, is a much diminished inclination for the creaming of fat globules. Homogenization today is generally a two-stage process. The principal stage involves pushing the milk through a little decreased cylinder or pore. As the width swells and the milk flow becomes consistent, the pressure develops and fat globules break apart in the turbulence. The higher the weight, the little particles. What amount of pressure? Actually 2000 to 3000 pounds for every square inch. Albeit some superhomogenizers work multiple times the air pressure, 14,500 psi and higher. You can stick milk through entirely little openings with force that way. Before homogenization, fat globally go in size from 1 to 10 microns. Afterwards, the size is decreased to 0.2 to 2 microns. As the little fat globules start to form all over again, they incorporate into pieces of Faye and casein in their seeds. Some are totally encompassed by some layers of proteins molecules. The reason is for these new artificially adjusted globules to cluster together. Stage 2 of the homogenization procedure separates every unwanted cluster and ensures everything remains in synchron. From what we have seen thus, featured tell that the purification of milk before consumption is key and more importantly vital to our health. Homogenization, breaking cells and tissues. The first step in the purification of most proteins is to disturb tissues and cells in a controlled fashion. Using gentle mechanical procedures called homogenization, the plasma membranes of cells can be ruptured so that the cell content are released. For commonly used procedures are shown here. First, break cell with high frequency sound. Use a mild detergent to make holes in the plasma membranes. Third, force cell through a small hole using high pressure. Fourth, shear cells between a close-fitting protein plunger and the thick wall of a glass vessel. Cell suspense or tissues. The resulting thin soap called a homogenate or an extract contains large and small molecules from the cytosol such as enzymes, ribosomes and metabolites as well as all the membranes bounded organelles. When carefully applied, homogenization leaves most of the membrane's bounded organelles intact. Next is centrifugation. Centrifugation is the technique which involves the application of centrifugal force to separate particles from a solution according to their size, shape, density, viscosity of the medium and rotor speed. Not only is this process used to separate two miscible substances but also to analyze the hydrodynamic properties of micromolecules. More dense components of the mixture migrate away from the axis of the centrifuge move to the outside, while less dense components of the mixture migrate towards the axis, move to the center. Chemists and biologists may increase the effective gravitational force on a test tube so as to more rapidly and completely cause the precipitate to gather on the bottom of the tube. The remaining solution may be discarded with the pipette. Centrifugation of protein solution, for example, allow elimination of impurities into the supernatant. There is correlation between the size and density of a particle and the rate that the particle separates from a heterogeneous mixture when the only force applied is that of gravity. The larger the size and the larger the density of the particles, the faster they separate from the mixture. By applying a larger effective gravitational force to the mixture, like a centrifuge does, the separation of the particles is accelerated. This is ideal in industrial and lab setting because particles that would naturally separate over a long period of time can be separated in much less time. The rate of centrifugation is specified by the angular velocity, usually expressed as revolutions per minute (rpm), or acceleration expressed as the conversion factor between rpm and g depends on the radius of the centrifugation rotor. The particles' settling velocity in centrifugation is a function of their size and shape. Centrifugal acceleration, the volume fraction of solids present, the density difference between the particle and the liquid, and the viscosity. The most common application is the separation of solid from highly concentrated suspensions, which is used in the treatment of sludge for debating where less constant sediment is produced. In the chemical and food industries, special centrifuges can process a continuous stream of particle-laden liquid. Centrifugation is the most common method used for uranium enrichment, relying on the slight mass difference between atoms of U-238 and U-235 in uranium hexafluoride gas. Differential centrifugation. Repeated centrifugation at progressively higher speeds with fractionate cell homogenates into their components. Centrifugation separates cells components on the basis of size and density. The larger and denser components experience the greatest centrifugal force and move most rapidly. They segment from a pellet at the bottom of the tube, while smaller, less dense components remain in suspension above, called the supernatant. Low speed centrifugation, supernunt, supernunt 2, supernunt 3. Medium speed centrifugation, high speed centrifugation, very high speed centrifugation. Cell homogenate, pellet 1, whole cell nucleic cytocleitos. Pellet 2, pellet 3, pellet 4. Isolation and purification of biomolecules. Concept media. Selection of our product range for isolation and purification. The more gently the cells are assayed, the more intact the biomolecules will remain, suitable for manual and automated preparations. Isolation of biomolecules or organelles. Homogenate, pellet, supernunt, pellet 2, pellet 3. Purification methods. Thin layer chromatography. Thin layer chromatography (TLC) is a chromatography technique used to separate non-volatile mixtures. Thin layer chromatography is performed on a sheet of glass, plastic, or aluminum foil, which is coated with a thin layer of adsorbent material, usually silica gel, aluminum oxide, aluminium, or cellulose. This layer of adsorbent is known as the stationary phase. Gas chromatography (GC) is a commonly type of chromatography used in analytical chemistry for separating and analyzing compounds that can be vaporized without decomposition. Typical uses of GC include testing the purity of a particular substance or separating the different components of a mixture. In some situations, gas chromatography can be used to prepare pure compounds from a mixture. Electrophoresis. Electrophoresis is the motion of depressed particles relative to a fluid under the influence of a statically uniform electric field. Electrophoresis of positively charged particles is sometimes called cataphoresis, while electrophoresis of negatively charged particles anions is sometimes called anaphoresis. The electrokinetic phenomenon of electrophoresis was observed for the first time in 1807 by Russian professor Peter Ivanovich Strachov and Ferdinand Friedrich Russes at Moscow University, who noticed that the application of a constant electric field caused clay particles dispersed in water to migrate. It is ultimately caused by the presence of a charge interface between the particle surface and the surrounding fluid. It is the basis for analytical techniques used in chemistry for separating molecules by size, charge, or binding affinity. Determination of structure. Elemental analysis. Elemental analysis is a process where a sample of some material, soil, vast or drinking water, bodily fluids, minerals, chemical compounds, is analyzed for its elemental and sometimes isotopic composition. Elemental analysis can be qualitative and it can be quantitative. Elemental analysis falls within the ambit of analytical chemistry, the set of instruments involved in deciphering the chemical nature of our world. Spectroscopy. Spectroscopy is the study of the interaction between matter and electromagnetic radiation. Historically, spectroscopy originated through the study of visible light dispersed according to its wavelength by a prism. Later the concept was expanded greatly to include any interaction with radiative energy as a function of its wavelength or frequency, predominantly in the electromagnetic spectrum, although matter wave and acoustic waves can also be considered forms of radiative energy. Recently, with tremendous difficulty, even gravitational waves have been associated with a spectral signature in the context of the Laser Interferometer Gravitational Wave Observatory and Laser Interferometry. Spectroscopic data are often represented by an emission spectrum, a plot of the response of interest as function of wavelength or frequency. UV-IR spectroscopy is a technique that complements the structural elucidation of molecules. NMR is nuclear magnetic resonance spectroscopy, most commonly known as NMR spectroscopy or magnetic resonance spectroscopy, is a spectroscopic technique to observe local magnetic fields around atomic nuclei. Mass spectrometry (MS) is an analytical technique that measures the mass to charge ratio of ions. The results are typically presented as a mass spectrum, a plot of intensity as a function of the mass to charge ratio. X-ray crystallography (XRC) is the experimental science determining the atomic and molecular structure of a crystal, in which the crystalline structure causes a beam of incident X-rays to diffract into many specific directions. Functions and metabolism can be studied by intact animals, whole cells, tissue homogeneous, isolated cells, organically, metabolites, and enzymes. Summary: we have discussed in this class isolation of biomolecules, centrifugation, products. Thank you. Have a good day.